The year was 1996 when all the world's attention turned to a particular sheep. In that year, Dolly, the planet's most famous sheep, was introduced to humanity as the first successfully cloned mammal. A clone was created. Dolly was the perfect and woolly copy of another identical sheep. She was born in Scotland in July 1996 and died 6 years later due to a lung disease. Her brief journey on Earth, however, left an unparalleled legacy for modern science. Dolly proved that life could arise from other forms, surpassing the natural process of the union of two cells. A single cell from any part of an individual's body was enough to generate life. Since then, researchers have refined the technique and made advancements in embryonic research, yielding significant results in various scientific fields. Now, a new clone, this time of a monkey, once again captures the world's attention. Human cloning, in turn, is once again placed on the table for ethical and moral debates. Earth echoes through the fascinating and microscopic universe of cells, the smallest structural units of all living beings. Their resonance, however, reverberates grandly, being the main witnesses to the wondrous miracle of existence. In 2018, Chinese scientists announced the birth of the first cloned monkeys using the same technique as Dolly the sheep. That is, conception was carried out from a single adult somatic cell. Somatic cells are any cells responsible for the formation of tissues and organs in multicellular organisms. Before Dolly, the scientific community believed that cells, once they had committed to a function, could not assume a new role. With the cloning of the sheep, it was proven that a cell with a specific function could be reprogrammed to act as a reproductive cell. Reproductive cells are responsible for giving rise to other cells until the complete formation of a new living being. Since then, the method has been tested in 23 different species. However, until 2018, scientists had not been able to manipulate the biology of monkeys, which are closer to humans. The experiments resulted in spontaneous abortions or clones with deformities. The first cloned monkey in history, named Tetra, was born in 1999. It was created using a different technique that emulates the division of an embryo to create twins. In 2007, scientists in the United States also cloned monkey embryos but not live animals after gestation. Indeed, it was in 2018 that the Institute of Neuroscience at the Chinese Academy of Sciences used the nuclear transfer technique. This approach allows the development of identical clones from an adult cell of a single individual. For this, researchers used fibroblasts, young cells in full productive activity, from a monkey fetus. The fertilized eggs were incubated in 2 adult females, giving rise to 2 monkey clones, named Zhong and Hua. Mu Mingpu, director of the Shanghai Institute and co-author of the research, emphasizes that the goal is the investigation of certain human diseases. The expert argues that many diseases, especially mental ones, depend on test subjects more similar to humans than rats. Many drugs that prove effective in controlling brain diseases in rats end up being inefficient in humans, with various side effects. In this sense, the director of the Barcelona Regenerative Medicine Center, Angel Raya, agrees with the Chinese scientists. The researcher points out that there are conditions, such as Parkinson's disease, that require the exclusive use of monkeys for experimenting with new drugs. Since then, Chinese scientists have been working on new techniques to clone chimpanzees, orangutans, and gorillas in the near future. Going beyond the study of genetic diseases, cloning could also be used for the conservation of endangered primates. The results of the research, however, reignite controversies and ethical debates throughout society. If there is a possibility of cloning primates, the idea inevitably extends to humans. This is considering the similarity of the genetic makeup. For example, chimpanzees share 98.8% of our DNA. Scientists believe that human cloning may be getting closer to reality every day. However, they also consider that society would not allow this type of scientific advancement. In January 2024, monkey cloning once again made headlines in major news outlets worldwide. This followed the survival of a Rhesus monkey clone for over 3 years, reaching adulthood. The achievement, documented in the journal Nature Communications, marks the first successful cloning of the species. The monkey, named Retro, was born on July 16, 2020. The team responsible for the cloning is the same as the one behind Zhong and Hua, both healthy and thriving at 6 years of age. To create Retro, Chinese researchers adapted the technique used in cloning the sheep Dolly. The new unusual procedure involves a kind of placenta transplant. This is because the standard cloning technique typically results in extremely low survival rates. The outcome in primates, especially, has been particularly limited in recent years. In 2022, Chinese researchers cloned a Rhesus monkey using the same technique, but the animal survived less than 12 hours. Now, with this new approach, experts have managed to reduce defects that could hinder embryo survival. At the same time, fewer embryos and surrogate mothers were needed to achieve the result. Rhesus monkeys are easily found in Asia, with populations in Afghanistan, India, Thailand, Vietnam, and China. Chinese experts prefer this species due to its genetic similarity to humans. They are tested in infection and immunity experiments, with great potential to accelerate drug testing. Furthermore, scientists use clones to model mental epidemics, such as depression and anxiety, affecting humanity globally. The effectiveness of medications like antidepressants in monkeys provides more confidence for application in humans. Chinese researchers claim that they intend to use as few animals as possible to prove the efficiency of medications. They note, however, that clone production still involves many substitutes, and the effectiveness of cloning has not been fully resolved. By definition, clones are artificial organisms that rely on significant technological intervention. Every clone is composed of a population of cells derived from a single cell, which in turn produces a genetically identical copy of the original. Once created, they will biologically continue to exist, with unique personalities, ideas, and experiences identical to a natural living being. The only difference is that clones have been genetically perfected. Cloning goes beyond mere experimentation with animals, expanding into various fields of science. Multiple benefits and advancements have been developed through cloning, not only for humanity but also for nature as a whole. In addition to reproductive cloning, science also explores therapeutic cloning, which does not involve the development of a new living creature. In this procedure, the nucleus of a cell is transferred to an egg without a nucleus, acquiring characteristics of a stem cell. Biomedicine utilizes stem cell clones from patients in research on regenerating various tissues, such as muscles, bones, cartilage, and nerves. Thus, individuals with traumatic injuries and organ degeneration could have tissues regenerated or receive transplants of cloned organs. Researchers also work with the method to study spinal cord regeneration in individuals with reduced mobility. Additionally, stem cells can replace neurons, heart cells, and insulin-producing cells. This research opens new avenues for treating serious diseases such as Alzheimer's, heart failure, and diabetes. Beyond the field of medicine, genetic engineering also applies cloning in farming. In this segment, animals are modified to improve the quality of milk and meat, while plant cloning selects specimens more resistant to diseases and pests. On one hand, reproductive cloning raises ethical debates and legitimate concerns regarding animal rights. On the other hand, clones can be seen as true genetic archives of rare species and animals facing extinction. The fact is that these discussions evolve as technology advances. New revolutionary fronts emerge every day to enhance the well-being and evolution of living beings as a whole. Under the microscope, we are reminded that greatness lies not only in tangible things but also in universes invisible to human eyes. Cells, these minute architects of existence, ceaselessly construct life and persist in this grand saga of survival. Where there is life, Earth echoes will continue resonating, bringing knowledge and scientific advancements. If you enjoyed this content, don't forget to like and share the video. See you in the next Cell Division.